Hello friends, welcome to my channel Plant Science for You. Today in this video we are going to talk about Anammox, that is anaerobic ammonium oxidation. We are going to see about its origin or how it plays an important role in nitrogen cycle, its application in industrial purposes and its actual metabolism process we are going to see. So let's start. Before starting about the process, let's take an overview about nitrogen cycle. We all know that nitrogen is an essential nutrient for all living organisms. It is a component of our cell. It comprises different nucleotides, protein, etc. And we all know that nitrogen is about 78% of our environment or our atmosphere. But due to its presence of a triple bond, we the living organism cannot break it down. Some microorganisms are there. They plays an important role in fixation of this nitrogen into the available form, like ammonia, nitrate, etc., which the plant takes up, and we, when the eats plant vegetables, we get those from the plants. This way, the nitrogen is getting assimilated in our body and plays an important role in our body structure. The nitrogen cycle describes all the natural processes by which nitrogen is transformed and cycled through various forms in the environment. That means the nitrogen is fixed in the form of ammonia, then it is converted in the form of nitrate, then it may convert into nitrite. Okay, in this way, it is going to cycle back to the original nitrogen form via different cyclic processes like nitrogen fixation, nitrification, denitrification, ammonification, and some other processes too, which we are going to see. After that, if we see what is the nitrogen fixation means the conversion of atmospheric nitrogen into ammonium by different bacteria and other organisms too. After that, like ammonification, what we say that it is the conversion of organic nitrogen compounds like protein, etc., to ammonium by decomposers. After that, likewise, the above two process describes about the how the ammonium is assimilating in the soil. After that, if we talk about the nitrification, it is the conversion of ammonium into nitrate by bacteria, and nitrite is an intermediate step in the process of nitrification. After that, we have seen so far the nitrogen is getting assimilated in the form of ammonium and nitrate. Now, this is the half of the cycle. After that, how this ammonium and nitrate is cycled back to the atmospheric nitrogen, we can see there are two players. First one is the denitrification, where the nitrate to nitrogen gas is converted, leading to the release of nitrogen back to its atmosphere. The most recent discovered bacterial process done by some specific Anammox bacteria, which done anaerobic oxidation of ammonia, which convert ammonium to nitrogen gas using nitrite as an electron acceptor, which we are going to see in the next part. Here we can see the cycling of the nitrogen. After that, as we have talked till now, that both the processes, denitrification and the Anammox, involves or plays an important role in the release of the nitrogen gas in the atmosphere from the ammonium or the nitrate. But there are some certain differences between them. That in case of denitrification, we see the organisms involved about a variety of bacteria, including Pseudomonas, Paracoccus, and Theobacillus. Whereas in case of Anammox, the specific group of bacteria which called as Anammox bacteria, such as Candidatus, Brocadia, Anammoxidans, and Candidatus Kuenenia, plays an important role in this conversion. That if we see what is the substrate used by them, the substrate used by the denitrification bacteria is the nitrate, whereas Anammox bacteria use the ammonium and nitrite as substrate. Oxygen requirement. Denitrification is mainly in an aerobic process, and it is undergoes under microaerobic condition or even anaerobic conditions too. But Anammox is wholly a anaerobic process. End product, if we see, the denitrifications convert nitrate into nitrogen gas, whereas Anammox convert ammonium and nitrite into nitrogen gas. In both cases, the end product is the same as the nitrogen gas. But the efficiency, if we talk about, the denitrification efficiency is less. Whereas an Anammox, there is a more efficient process compared to denitrification, as it requires less organic matter and energy to convert nitrogen compounds into nitrogen gas. Okay. Now let's discuss about Anammox a little more detail. If we see about the Anammox, it plays a very important or significant component of biological nitrogen cycle. After that, if we see about the discovery of the Anammox, it was discovered about 20 years ago in an yeast factory. Okay. Globally, if we talk about 30 to 50% of the nitrogen gas is produced in the ocean. Mainly, this process is going in the ocean or marine environment. About 30 to 50% nitrogen gas is released in this form. The Anammox bacteria are obligately anaerobic and also the chemolithoautotrophs, and they belong to the phylum Planctomycetes. There are 5 Candidatus Anammox genera are identified so far, like Kuenenia, Brocadia, Anammoxoglobus, Jettenia. They are all freshwater species, whereas Scalindua is a marine species. These are all the important genera which we have discussed. After that, if we talk about some other features of Anammox bacteria, like Anammox bacteria can grow from -2 to 43 degrees centigrade. That means there is a various range of or tolerability in them to tolerate different environmental conditions, different temperature conditions. Whereas if we talk about the pH, they prefer something like neutral to some alkaline pH. But the optimum, if we talk about 8, while the optimum temperature varies with different species, as we have talked. These bacteria have a very uh, long doubling time. That means they grow very slowly. They grow very slowly. That means their doubling time takes about 10 days to 22 weeks. And if we talk, there is a important compound or metabolic intermediate, we can say, that is the hydrazine. You all know that it is one of the strongest reducing agent in the biological system. It is produced by this type of Anammox bacteria. And these Anammox bacteria cannot be successfully grown in the pure culture. So, because due to their slow growth rate, uh, what we can do, we can use a sequence batch reactor to culture those Anammox bacteria in large quantity. This is an image, transmission electron microscopic image of the Anammox bacteria, which is Kuenenia. After that, if we talk about the cell biology, the biology of the cell, that is, if we see the inner structures of the Anammox bacteria, we could find there is a compartmentalization. There's 3 types of compartment are present there, about which we call as a periplasm, riboplasm, and another one is the Anammoxosome. In them, the Anammoxosome is the most important one, which we will discuss. If we see that the Anammox bacteria exists in the coccoid cell structure and the diameter of 800 to 1100 nanometer, they have the unique cell wall composition. If we see their cell wall composition, they lack peptidoglycan and outer membrane, that means they are something distinct from gram positive and gram negative bacteria too. And if we talk about their membrane lipids, that means the cell membrane, we will see there are also an important features like they are both ester linked and ether linked. We know that the ester linkage is found in the bacteria and ether linkage is found in the archaea, but both of them is found in this case of Anammox bacteria. Okay. And another most important lipids is found in the Anammox bacteria, known as the ladder-den lipid. Why this name? The ladder, there is a ladder-like structure, like here you can see the ladder-like structure is found in them. That is why we call it as a ladder-den lipids. It is found in the Anammoxosome and are unique to Planctomycetes, the phylum of the Anammox bacteria. As we have talked that the Anammox bacteria consists of the three compartments like periplasm, riboplasm, and Anammoxosome. If we go in little more detail, we can find this is the Anammoxosome, this is the riboplasm, and this is the periplasm. Okay, if we go little bit detail about them, we can see that the Anammoxosome is about 50 to 70% of the total cell volume covers, and it is actually act as a mitochondria like processes. All catabolic processes is undergoes in this Anammoxosome. It is more like eukaryotic mitochondria, and it possesses as the we can see the mitochondrial structure. There is a many invagination is there. So here is also why this invagination you must know that if there is an important functions to do like DNA, which is highly supercoiled in structure. So why this supercoiling? Because there is a most important information is there. Likewise, in mitochondria, the inner membrane is folded and like invagination is made. That means it is very important to them. So as like that, the Anammoxosome is most important compartment for Anammox bacteria because the main metabolism processes occurred there. So there we can find there many invaginations. After that, the metabolism is very slow, which we have talked. The hydrazine, which is produced in the Anammoxosome, is readily transverse biological membrane and must be kept inside the Anammoxosome. So, as we have talked, that it is a strong reducing agent. It can damage the DNA. So, it is important to keep in this side in the Anammoxosome. Okay. And we have talked that if this is the Anammoxosome, there is a ladder-den lipid which provide this protection. The ladder-den lipid in the Anammoxosome membrane decreases its permeability to proton and hydrogen. That means this cannot go outside because in the riboplasm, mainly the translation or transcription processes undergoes. So it should not be go outside. It should remain in the Anammoxosome, and the ladder-den lipid in the Anammoxosome membrane plays the important role in keeping those inside. After that, if we see the riboplasm, it is the cytoplasmic compartment-like structure, and it possesses the nucleoid and ribosomes, and it is the site for transcription and translation, and it is also stores the glycogen. After that, if we talk about the periplasm, it is the outermost cellular compartment. If we see this one is the outermost part of the compartment, and ladder-den lipids in the cytoplasmic membrane provide an additional strength to it. The role of periplasm and whether it functions of periplasmic or of cytoplasmic space is still a subject of research. That means whether it has the role of doing the periplasmic space, as we have seen in case of bacteria. There is a cell wall, there is a cell wall, and inside them there is a plasma membrane, and between them there is a periplasmic space. We don't know if it is the same role of this or not. After that, if we see the molecular processes of the Anammox reaction, we can find there is a this is the overall reaction we can see where the ammonia and nitrite is converted into nitrogen gas, which goes to the environment. If we broke down the reaction, we can find the first reaction occurs where the nitrite uh, is converted into nitric oxide. Then this nitric oxide is with ammonium is converted into hydrazine. After that, in the third step, if we will find that the hydrazine is converting into the nitrogen gas. So in these three different stages, there are three different enzymes which plays important role in conversion of this nitrite and ammonium into the nitrogen, which we will see in the next slide. Till now, we have talked about the internal structures of an Anammox bacteria, where we have seen that the innermost compartment, that is the Anammoxosome, and after the outside there is a riboplasm, and this metabolism process is all we will all see that this process is concerned about the Anammoxosome. If we talk about, if we see the Anammox, this is the, suppose this is the Anammoxosome inner compartment and this is the riboplasm. And this is the Anammoxoglobus membrane where the lateral lipid is found. If we see the metabolism process, in the first step, there is a reduction of nitrite into the nitric oxide. There, the important step, the first step, where the enzyme involved is the nitrate reductase. After that, the with the nitric oxide, the ammonium ion is combined and from the hydrazine is the second step. The involved enzyme is the hydrazine hydrolysis, which form the hydrazine. In the third step of the reaction, or which is the final step, where this hydrazine is converted into the dihydrogen gas in the third step with the help of the enzyme, the hydrazine or hydroxylamine oxidoreductase. Then if we see the electron flow in them, we can see that the from the hydrazine hydrazine to nitrogen conversion in the third step, the 4 electron is converted to is transferred to the cytochrome. Then it is going to the quinone complex. After that, it is supplied to the cytochrome BC1, and the cytochrome BC1 has an ATPase activity, which helps in production of ATP. And from the cytochrome BC1, the electron is coming to the cytochrome, and this cytochrome is then donating their electron to the two different enzyme, that is the uh, what we say nitrate reductase, and another one is the hydrazine hydrolysis. If we see that the one ATP is formed, now this ATP is further used for carbon dioxide fixation in this kind of bacteria. As we have talked in the earlier, there is a chemolithoautotroph. That means autotrophic significance is there because they can fix carbon dioxide, and using this ATP they can fix this carbon dioxide. So if we see take an overview of these, the three enzymes are involved in the three different stages. The first one is the nitrate reductase, which convert the nitrite into the nitric oxide. Then this nitric oxide and the ammonium combine to form an hydrazine with the help of the enzyme hydrazine hydrolysis. And then this hydrazine is converted to the nitrogen gas by the help of the enzyme hydrazine oxidoreductase. In this process, the 4 electron comes to the cytochrome, then it comes to the quinone, which is located in the lumen of the Anammoxosome membrane. After that, sorry, not lumen, in the membrane, Anammox membrane, there is a present of quinone. From the quinone, that is goes to the cytochrome BC1, and then the cytochrome BC1 it goes to the cytochrome, and this cytochrome gives the electron to the enzymes 1 and enzymes 2. In this process, the one ATP is formed, and using this ATP, the Anammox bacteria can produce or fix carbon dioxide. Okay. Now, if we see what are the effectors of metabolism of this Anammox metabolism, The first effector is the pH. There we have talked there is a need of neutral to alkaline, near alkaline pH. Uh, if this is hampered, some metabolic processes may got hampered. So the pH is one of the most important factors. After that, if we see the temperature is about 30 to 43 degrees centigrade. Then the most important one is the oxygen. Yeah, which we have talked that is an anaerobic process. That means the oxygen must not be present. So the dissolved oxygen concentration about over 2 micromolar can be reversibly inhibit the process. After that, if we see the light, light can actively decrease about 15 to 20% of its activity, because a light is not a direct effector of this process, but it can indirectly affect the process, because light can be the source of energy for different kind of alga or different types of phototrophs which can grow up, and they can suppress this kind of process or this kind of bacteria. So light is also affecting this process. After that, if we see the COD, that is the oxygen demand. This is the about if it goes above 150 milligram per liter, it can limit this activity. This actually represents about the organic matter. Okay, this is the chemical oxygen demand. After that, if we see the phosphate, it also acts as a limiting factor for the growth of the Anammox bacteria. Then we see the ammonium and nitrite. These two are the substrate of the Anammox bacteria, but above the certain level, they can decrease the activity of this Anammox bacteria. Likewise, in ammonia, this is like above 100 milligram per liter, which decrease the activity about 80%. And if you see the above 11 microgram per liter nitrite, which can decrease this activity about 50%. So these are all the factors which are affecting the processes in uh, animals. Now, if we see what are the actual application in Anammox bacteria, uh, if we talk that Anammox process is an important process of in nitrogen cycle. But in case of agriculture, there is a less importance, because if we apply the nitrate fertilizer or ammonium fertilizer, it may goes evaporates in the form of nitrogen gas, and that would not be favorable to us. But if we see in case of rice, where there is a 15 to 20 centimeter stagnant water is there, in those cases, this nitrogen uh, evaporation is plays an important role. But more efficiently, this Anammox process is important in case of marine bacteria, where the ammonium is converting into the nitrogen gas. And if we talk about their most important process in case of industrial purposes, they can be found as a wastewater treatment, or treatment of the sewage and wastewater treatment. Where there is mostly present the ammonia, this can be converted into nitrogen gas easily. And this process will not involve any greenhouse gas production, and less energy consumption is there. So it is very important aspects. And if we see this is mainly used in case of treating the waste laden waters, and including the sewage, if effluent, food processing plants, etc. This Anammox reaction can be coupled with aerobic oxidation of nitrate to nitrite, may achieve the complete oxidation of ammonia to dinitrogen, and this is desirable in case of wastewater treatment. And this coupling reaction can be implemented using either one vessel system or two vessel system to carry out those reactions. Like first is the Anammox reaction, and another is the Anammox reaction is basically the anaerobic reduction, the anaerobic oxidation. But the aerobic oxidation, there is a denitrification, there is nitrate to nitrite, then it goes to the nitrogen. These two processes can be coupled, using this one vessel and two vessels. Now, the use of this Anammox bacteria in wastewater treatment can lead to the significant cost saving. Environmental benefits are also there, as we have talked. They will require reducing energy consumption, reduce the energy consumption, and minimize the use of different chemicals too. So that's why this process becomes more important in case of industrial purposes. So these are the references I have used in this presentation, and thank you for watching. Like, if you like the video, please like, subscribe and share with your friends. Thank you.